we address a problem or a clinical problem in blood transfusion. So whenever there is a blood transfusion case, so blood bank should define the ABO and rhesus blood group, and the matching blood group should be transfused to the recipients. So, however, there is a type of rhesus called D variant, which is very hard to detect. And if this is the case, then there is a fetal complication whenever a mother who have had a blood transfusion before gets pregnant, then, and the blood transfusion was, what I mean, the rhesus was wrongly misreported and transfused to the mother, so the baby, the fetal complication will be the outcome. So this is one of the limitations in the traditional blood bank tests that's going on so far in many countries. So what is the current updates? Of course, in Western countries, to overcome this problem, a lot of DNA testing is done. However, this DNA testing is a 3-step process. So the step 1 is to take the blood and remove the DNA from the blood, which takes about 2 to 4 hours. And then the DNA copies will be made, like 1 million or 2 million copies, in order to be studied. And then the gene change or gene mutation study will take place. So all of these 3 steps, though they are very specific and sensitive, but they are like time consuming and they are quite costly. So what is our proposed study is to combine all these 3 steps as one step. And so far we have been successful in doing that, and we have been able to make 1 million copies of DNA without doing any extraction method. So it's like a direct from the dry blood sample. So we have collected blood samples from Oman, where I live, and we know that Omani population have different D variants, and which makes them very good candidates to do this project. So by the end of this project, we hope that we make a protocol, the first protocol in Oman to detect D variants, and of course a 1-step method which can be beneficial worldwide. Thank you so much.
Thank you, Mustafa. Very important work by the sounds of it. Do I have a question?
Yes, please.
Usually for blood transfusion to
Yeah.
avoid all these complications,
Yeah.
you do cross-matching between the patient blood or the recipient
Yeah.
blood and the donor blood.
Yeah.
Even this cross-matching will not be able to avoid or protect.
But once the antibodies are produced, that's it. So you want to avoid antibodies production. So if the wrong blood has been given, then antibodies will be produced. Then you cannot help it. So the aim of the blood bank is to stop these antibodies production. Okay?
Thank you very much.
Thanks.